Okay, today we have Alejandro Mieles, who is a postdoc working in the Global Change Group under supervision of Ana Terese. So today he's going to present a bit of his research that was already presented in an international conference funded by the Maria de Maetu Travel Award. So thank you very much, and the step is yours.
You're welcome. I'm super happy to be here because usually this is my third time here presenting my results. This is the first time there is a lot of people here. The other time it was only 4 or 5 people, so now I'm super happy to be stay here with a lot of very interesting persons because I really like to talk about Galápagos Islands. This is what my friends knows. I like to make presentations to talk about the islands, talk about the introduced species or chemical ecology. We was my first presentation, but unfortunately nobody was here, and I was so sad. But now, finally, I can share with you a very short presentation for you. Basically, this is we're talking about this island. This is Daphne Island. Well, first of all, it's important to mention that in natural science, most of the scientists usually are trying to figure it out how nature is working, how the ecosystem works. So, for instance, when we are working in our thesis, usually we try to make a framework with a lot of subjectives. We try to consume all the work, but basically we never get going for the final result that is trying to figure it out. Oh, but that's why most of the scientists usually focus in only one kind of research. For instance, research working with pollinators, or they working with herbivory, seed dispersal, etc., etc. Well, the idea is try to join all these interactions to try to figure it out how the ecosystem works. But this is super hard, actually. This is a big deal for natural science. So try to understand this complexity in the nature is super difficult, but philosophy in 15 years ago, this is about 2014, 2015, proposed a kind of different analysis to try to join all these interactions separately, try to join just one analysis. But the first publication basically that talk about the how ecosystem works using a different layers, it was in 2016 maybe. But that's why this recently approach for this multi-layer network analysis. So the thing is how to understand these interactions. If we study these interactions in a, I don't know, in a tropical ecosystem or Nearctic ecosystem is complicated because the functions, different factors are a lot of information. So that's why. But islands are particularly interesting because this is a closed system. For instance, in Daphne, who is a very famous island. I don't know if all of you knows why is famous Daphne. Well, Peter and Rosemary Grants, they are like a super rock star in science. They were working in this island, very small island, for more than 50 years, and they were living in this island for 4 years. Why is particular? Because this is basically a hell in this island. It's super hot, very small, no trees, no water, but they were living, and they educate the child in this island. It was fantastic. And also, this is the first experiment that they carry on for more than 4 years. Try to see what happened in an ecosystem, especially try to see if the evolution is passing during a very short period of time. Basically, when they published this book, they discovered, well, they publish the results that they noticed that some species, especially the finches, they changed in only one generation of the human. In 4 years, they studied in 1973, and they finished 5 years ago. Another all scientists, but basically they stayed during the big dry season in 1977, and they discovered the evolution forced one species of the finches to change, to make more robust and big, because the only food resources in this island was a very, very rigid seeds, and they changed, they modified the beaks to try to broke these seeds and consume something. And then in the El Nino event in 1982, this pattern changed and the pigs reduced again. That was fantastic, actually. That was why they are super famous. Also, Martin Wikelski, another scientist, studying a different island, the same pattern was changed during the dry season or El Nino season. So in this other research, Martin Wikelski, it was measured the size of the marine iguanas, and during the dry season, the iguanas were long, but during the El Nino, they reduced the size. That was fantastic, actually. That's why this is a study, these changes in the islands is super, super crazy. So other things that happened in this island was a new, what is the laser here? Here. So this was arrival for one big bird. This is coming from the big island in Santa Cruz. This bird flew for Daphne, and they produce a new generation of the individuals. They change the behavior, they change the songs and the behavior of other group of birds. That's crazy. Imagine that this is they publish these changes, this evolution in the real time. So when the group of anatravers select these islands, well, they were thinking to try to see something similar that the grants study. So this is the real condition working in this island, because there is no trees in this island. So we use caves and cooking in these caves and just prepare some tents. Basically, it's super hard. So this challenge in our research, for instance, again, is trying to see all these changes, all these interactions in a real time, but using a different factors like pollinators, seed dispersal, herbivory, or even fungal association with plants. So studying the multiple interaction types at the same time, again, this is one challenge. That's why we were in this island, detecting and quantifying links across the layers and developing a new dynamic connections models. So this is in a large frame for this big project. The Anna Travis group, they were working in different islands, in the different islands around the planet, including Seychelles, the Arctic in Svalbard, what is the other one? Oh, this is the Canarias and also Galápagos Island. But recently, last year, they included a new archipelago. Well, a new archipelago is in the Tierra del Fuego in Chile, to try to cover more of the island along the different degrees in the world. So they have, I say they have because I only included in this project just recently. So they have three main goals for this: is to build a multilayer network in oceanic and pristine islets involving the key ecological functions. So the additional of this, the idea is to try to compare one pristine island and the other with disturbed island, but this is not included in this presentation. The second goal is to identify plant species that mostly contribute to the multilayer network cohesiveness. This is a word to pronounce. And the third one is to assess which are the main phytocentric ecological function contributing with the whole network structure. Obviously, this have a particular methodology for this. This is including monitoring a transit 100 meters per 2 meters way and covering whole island in this small island, because this is relatively a small island. Well, some part is difficult to walk, but even consider these difficulties for walking on this island, we cover most of the island. Even the hardest part, we were working and we collect information for this island. So the target for this methodology was, sorry. Is pollinators, as again, herbivory, seed dispersal, soil regulation, analyzing the nematodes in the soil, and root associated with fungi. The idea was collecting or notice the information for who pollinators going for which species of plants or what kind of herbivores feed on plants or especially plants or seed dispersal. The idea was to analyze the fecal material for birds and lizards and try to count the seeds in the fecal material. And so regulation is you was only collect soil for 250 grams of soil near to the species of plants and try to see what is the nematode species are sharing the space in the soil. Finally, we try to compare the dry season versus a very rainy season because the same situation at the grants, the idea was try to see is a different factors are forcing the changes in the ecosystem. So 2023 was a particular year because it was not basically a very dry year, but the conditions are still dry. But 2025 was particular rainy season. It was raining for a lot of time, and actually the plants species were present in this island for more than 4 months, which is very rare because in this island, basically we can see 5 or 6 species of plant. One is the cactus, and other are trees that they don't have leaves, and other more like a brush. They are permanent plants in this area. But in the rainy season in 2025, there was a lot of species of plant that were present, including also grasses, some 3 species of grass. So to collect information for the fungal groups, what the idea was to collect the roots material in every single species of plant, and we're using a metabarcoding analysis to try to analyze which species of fungi are associated with the plants. And then we classify in 3 different groups, and we analyze separately the 3 groups of fungi. One was saprotrophic fungi associated with the decomposition, mycorrhizal fungi associated with nutrient uptake, and pathogenic fungi associated with fungal pathogenicity. So also we try to including a different kind of analysis, like a bipartite network metrics, including specialization, interaction evenness, connectances, nestedness, and robustness. And also we make a multilayer network metrics. So I want to go into this table, just mention a couple details for this. This is for the previous analysis for this. This is the table, but basically I can say this network has a high speciality, high interaction evenness, and high robustness. The only difference was in connectances. This is real clear because most of the network is more specialized species of animals or fungal to species of plants. That's why this is the difference. So this is the more beautiful result at the moment. Is basically what we try to do is connect all these different functions, 7 different functions, in one multi-layer network. If we see here species like Opuntia is a cactus endemic from Galápagos, or Croton scoleri is a small brush. These 2 species are like separately because it's interesting because all these 2 species you can see along of the year, and they support a very dry season. Even they can stay for the island for more than 4 or 5 years without receiving water. Also, Another species is the subium, is succulent plant, and the other is what is the one? Sorry. Well, Sida is another species, but this is separately because we only reported this species of plant during the rainy season. Tribulus and Sesubium, they're also a permanent species, but there is the number of plants is very low. So in terms of the contribution for every node these plants, we can see pollinator. We recorded 51 species, pathogenic fungi 73, seed dispersers 6, nematodes 32, saprotrophic fungi. It was the more largest number of species was under 167 species. Well, this is not a species. It's basically just a code for identify different groups of fungi. Symbiotic fungi was 10, herbivory 67. And in a total, we recorded 26 species of plant, but we don't detect different functions along the all year studies. We discard 4 of these, and we only keep with 22 species of plants. So the modularity here is interesting because if we separate the results in terms of nutrient uptake, dispersion, herbivory, etc., we can see nutrient uptake has a strong cohesion of the network. That means this is very specialized factors and functions that connect with just one function with one species of plants. But dispersion is very low. That means we can see different species of plants sharing with different species of animals, which is very interesting too. But basically, in general terms, the modularity of this network is more specialized compared to the other network, like in the mainland or in the tropical forest. So this is the result for the goal 2. Is basically is more focused in the keystone species. So the idea is here is show what is probably the keystone species, and we can see this is basically 5 of these species of plant. So which is interesting is these 5 species of plant. 4 of them they're like a permanent species that we can record even in the dry season or in the rainy season. So for instance, Pseudobutyllum, the pauperatum is important for pollination and herbivory, but this is the only species of plant that we can record it during the rainy season. Portulaca is another succulent plant. It's important for dispersal. So a lot of seeds for these plants we recorded in the fecal material of the pigeons endemic for the islands, and also for pathogens. So this Portulaca is very interesting species of plant because it's a small one and produce a very beautiful flower. It's a yellow flower, but we only see these flowers after 2 months the rainy season. So if there is no rain during the 1 year, there is no flower for this plant. So Opuntia, Croton, and Tribulus, there was a permanent species of plant that we can record along the year. These 3 species were present in multiple modular layers, like herbivory, pollinator, and mycorrhizal. So in terms of keystone functions, the more important function was herbivory. So pollinator. This was most of the interaction between animals or fungi were associated with the herbivory and pollinator. So we can see the different modular layers. Is the composition is the highest, and the lowest was seed dispersal. Because well, this has probably a potential explanation. So when we collect fecal material for the pigeons or finches usually they destroy the seeds. It was very difficult to identify the species of plants, but the pigeons they ingest all seeds or fruit, and the seed is more easy to identify. That's why it was probably the lowest number of seed seed identified in the fecal material was most of the finches, they destroyed the seed. Also, for Opuntia, Opuntia is a relatively large seed, but even when there is no food in this island, the finches broke the seed and they feed on the internal part of the seed. So that's why maybe the seed dispersal is very low, but basically most of the animals are associated with to eat different species of seeds. So here is again just to make a more effort to try to say the most important plants basically were Opuntia is a cactus, Portulaca is a succulent, and Croton because they are the permanent species in this island. But in terms of function above the soil is the herbivory, and into the soil it was the pathogens associated. These pictures were taken during the rainy season. This is very rare because these species of moth we never recorded along the other years, or even this terraphoric micro Lepidoptera is also a new record for this island. So this is the kind of different seeds what we recorded in the fecal material. So when we compare the dry season versus the rainy season, we try to join different years, like 1999, 2018. No, sorry, it's 2018, 2019, and 2023. And then because there is very few information during this year, or there is no herbivory associated with plants, or there is no flowering plants. So even when the Anna Traverse group visited this island in 2 different seasons, it was very hard to collect information. But when we join all this information for the dry season in just 1 year, even comparing with the rainy season, it was up to the double of number of species that they have a new functions in the island. So the thing is, when we see here for herbivory, this is changed completely when the rainy season comes. So basically, there is more food resources. Species that only feed on one plant, now they feed on different plants, except for Opuntia or Distimaki. Opuntia is because it's a cactus. There's very few herbivores feed on these species. A couple, maybe grasshopper or curculionid beetle. But in Distimaki it was only because these species of plant are appearing during the very a couple of weeks, basically a few days or a couple of weeks, and then disappear. But also Distimaki is interesting because I don't know, maybe it's for morphology or ethology. The plants, the morphology of the plant, they have a hairy and a greasy material covering the leaves. So maybe that's why there is very few herbivores feed on this plant. But in terms of pollination, this is super important. So again, it's a pollinator. This is more like doesn't change more the pattern even during the dry season or rainy season. So this is just information for you. Unfortunately, this is a very beautiful island, but in the past, in the Second World War, the U.S. Army they fire against the island, and you can find a lot of these castles, the bullets pieces, or even a bomb in this area. So why is this is interesting information for you? Is because if you see, it was in a rainy year, so we can see the soil. But when it's raining a lot, they remove the plants and we can see the bombs. They're active, actually. But nobody touched, because if you see this here, this is the trigger. If you touch this, probably explode. We don't know, but this has a 70 years. It's a real picture. We found this the last time we visited this area. So the changes here is amazing because this is a kind of relatively beautiful species of plant during the rainy season. But if we see, this is the historical picture for the Peter and Rosemary Grant. There was a dry season in different years. And this it was in a rainy season. It's super different, but the thing is, this is not every year change. It changed for decades, maybe 20 or 30 years, this pattern happened. So that's why this is a super interesting island to study in this place. And this is a nice view. Daphne Major is an island connected to the Santa Cruz Island, but coming from another peripheral volcano coming from the ocean. So this is why I have to present to you, and thank you very much. This is a project team. There is a lot of group of people working on this project, and thank you very much. You have question, please ask me wherever you want. Yeah.
Hello. That may be just a lack of my background, but I can't really get the concept of the ecological function proportions. So, I mean, how do you retrieve this score? Because to me it seems like, okay, you just define the some ecological functions when you consider it from the ecocentric perspective. They are less important than the others in general. But if you raise one function, I mean, the system will probably collapse anyways. So how do you account for that? And
Yeah.
to me, like the initial like starting point would be that each ecological function is like equal, because if you don't have any extra information on that.
Yeah. Well, sorry about that. Actually, I have this information in the paper that we are working on, but it's just no. Well, it was. I was not authorized to present this information, but I can say something about that. Basically, when we calculate this number that you mentioned, we have a graph, and this Mar, especially Mar, is a physicist. They was working trying to make a formula to try to calculate when the ecosystem collapse, consider the keystone species of plants or consider the keystone functions. So in both, it was different. For instance, for keystone functions, to try to preserve the ecosystem in this island, we were able to lose maybe 3 different functions. If we lost one extra function, the ecosystem collapsed. In terms of the species, it was very curious because the system still is function even if we miss 15 species of 22, which is very curious. Well, that's the number says. That's why I didn't present this, because it's probably a very problematic topic to talk about it. And maybe we need to discuss more about that. Why there is a large number of species of plant we need to lose to the ecosystem collapse. So that's why I know you mentioned this, but definitely it's complicated to talk about this in this special island. I don't know, maybe the system is different in a tropical ecosystem or in the Arctic ecosystem, probably it's different. But in this island, even if we lost most of the species, probably the ecosystem is still remaining and preserving some species of plant. This is the number that's said, but in real terms, we need to at least, I don't know, maybe 40 or 50 years more like at the grants to try to see or removing different species of plant in a controlled experiment to see what happened with this.
All right. I'll get back to the doctor. Still have some questions.
I have a question, maybe you haven't studied that, is about in To what extent marine resources around the island are used in the functioning of this community? Like, do any animals use food sources from marine origin? Or so to what extent the marine environment around the island supports the functioning of the ecosystem that is in the island, being a very small island?
No, we didn't study that, but actually, I know at the beginning, the group of, they were considering analyzing the fecal material for sea lions and also the seabirds. So this is also the fecal for the crabs, but there is no information about that. In the area where the crabs or marine iguanas or sea lions or even seabirds stay, there was very few fecal material. Also for marine species of the marine birds, they produce a kind of very wide. This is more calcium fecal. So I think probably we can compare this if we make a metabarcoding analysis for this fecal material, but we didn't consider this, unfortunately. There is a lot of information probably coming for this interaction between marine and land. Maybe in a different system, like I don't know, the other place in disturbance place is in the beach far from this island. The name is the Garrapatero. They have more connection with the marine and terrestrial. Probably we can see here more interaction that we can consider for the future.
I had a curiosity about the keystone species, because you said you found 5, right? I guess it comes from the abundances that you see there. But can you go back to the slide? Right. So I didn't really. It's pretty new to me exactly how you decide which ones are keystone. But if you just look at the abundance, to me it's not clear why 5 and not either 4 or 7, right? So there are 2 drops there, 2 big drops, and it's curious to me that that you decide to include the 5th, but for example not the 6th.
Yeah, you're right. Yeah, definitely. You completely. I completely agree with you. But the thing is, why we decide these 5 and not 7 or more, is because we run different models to compare these numbers. But the thing is, it was a decision more based on the presence of the species in the island. So that was why I mentioned several times these keystone species are present along the year. So that's why maybe and that's obviously we can discuss about that, but a species of plant like Pseudobutilum, we can see this species only during the rainy season. So why is including here? Because Pseudobutilum is a plant of species that we can see a lot of functions like in fungal or pollinators or herbivory. So and also the seed dispersal, probably I noticed in the soil a lot of seeds for these plants, but this is the seed that the finch has broken. So that it was not only a mathematical comparison for this keystone species of plant. It's more like the function into the system and what kind of functions are covering for the different factors that we are considering. This is a combination.
So what differentiates the 5th from the 6th?
Well, we can include 6 or 7. This is just a number. Yeah.
But I said, well,
yeah, we can say
at a certain point you're going to cut. Right?
Yeah,
Okay,
exactly.
So
Yeah.
based on which criterion?
Yeah, I said again, the criteria was what is the role in the ecosystem, basically.
Yeah, but that's not a criterion. Meaning, you want something quantitative, right?
Yeah.
Okay, so what exactly is this quantitative thing that you say, okay, I'm gonna draw the line here. This guy's gonna call them that, and these guys are not gonna call them that.
Yeah, well, again, this is just a combination of criteria. Yeah, they say probably we can include it more, but actually they are included in the manuscript we are preparing. But basically, we are describing what is the importance of this species of plant. Yeah, this is a lot of information we can say it's in the paper, but basically it's because. It's hard to say. For instance, this species, croton. Croton is here. It is not included in a keystone species here in red, but I mentioned in the here. So if you see, we can say 5 here, but actually I mentioned more than 5. Actually, I mentioned 7 probably. This is just the. It's just I market this because I like to talk about the first 5 species of plant, but also there are more. Actually, you're right.
More questions?
Another one. Um, I'm also working with, like, well, not with that amount, but different levels. And what I see is that may be a big problem that, for instance, for different players, the type of data is different, right? And sometimes you just grab the soil around the roots and you have like abundance of nematodes, right? You can transform it into biomass or whatever. Sometimes you have fungi, which is based on metabar coding with a lot of bias of this method itself. And then you try to combine it inside one system. So just what do you think? There some good way to tackle this heterogeneity of methods, and also, for instance, for some layers you collect the data which probably represents some area around a plant, right? Pollinators that is flying around, and some are just tightly associated with a plant. So, and then you, I guess, you somehow standardize this thing. Yeah, but so okay, I even don't know if it's a question or not, but what are your thoughts about this potential bias? Maybe you also see how we can kind of.
Yeah, actually, it's well in terms of how the group of collect information. This is complicated because, for instance, for metabolic coding to analyze the fungal associated with the roots. So the group only collect one time sample, so just in one period of time. So we have only this information because it's expensive to make more analysis comparing in different years, or I don't know, more number of replicants. So it's expensive. So pollinator herbivory functions we can access for the islands is just in one period of time, and even if this period of time is just a week ago or a week after, we can see different results because again I mentioned this portulaca species. They only flowering during the a very short period of time. So we can see other pollination associated with portulaca, but maybe there's a lot. Unfortunately, we can stay in this island for longer. We stay only for 3 days. One season, 1 year, and the other season, the other year. So try to collect a lot of information or all information we expect. This is basically impossible. So that's why you can see bias in the information, because obviously we can say the information is coming from the whole year.
We use standardized methods.
Exactly.
All the directions they have a way to standardize, and in the end we calculate the participation index in each function for each species. So everything is at least standardized by other we standardize every interaction, every type of interaction.
Doesn't then reflect the reality of the system. Maybe it's
That's
you cannot
impossible.
compare islands between.
But
each
this is
other.
the main
This is-
topic, kind of to join interactions that are different is hard. So we will this approach that was already published by Sandra, by the group. So the approach could maybe other approach can be developed. This is the a way that we now when we use to join all these kind of interactions. But
Yeah, but again, like what I'm saying, that maybe like if you have the same bias in each layer, we compare the two islands, so an island between each other is fine. If you want to have a good inference about the ecosystem itself, then you have problems again. No.
yeah, that's.
because-
They all the time they will be different. No, the fungi will never have a behavior like the pollinators because fungi are in soil and they are super
Yeah, but then.
abundant and different. But at least even for the keystone function plant species and also to use this participation index, the probability, and also we run new models to see how much these plants are important compared to different types of new models. And and also we compare this with abundance. We have the cover and abundance of these plants in the island, and we can see because we can expect that plants that are more abundant will be the most important to the system. But it's not really like that. The abundance is not correlated with the importance of the species. Sometimes some species are not so abundant and they have much importance to all the functions. So they participate in more function, even not being so abundant. So you consider these things, the abundance, the cover, the types of interactions we know that are different, but we process them in different ways and account for this difference. And when we can calculate the participation index, is a probability, we are accounting on that. So for each function, we put the interaction in the 0-1 metric to account for all these variables. We know that, but this is impossible to standardize this in a perfect way because there are different kinds of interactions. But if the system is like this, the nature is like this.
As you say, if you try to see a perfect representation of this is basically impossible. Even in this system, even in this closed system, it's very difficult because to do this is we need the same research like the Peter and Rosemary. Is stay for longer, collect a lot of information for years and years, spend a lot of money. This is very, very hard actually.
No doubt. This total thing for herbivory combination for different functions, that the total score when you sum up the keystone for each plant, does it originate from the combination of kind of some product or function from the abundance and participation of each plant? Is it like why are they different? What's the elements which define this total score for the function in this graph?
In this graph here or here?
No, well, in this right top or bottom graph. I know maybe Martin also. Do you understand the question?
See, but this is when you calculate the participation index for each plant in each function, and you have these metrics, and then you can calculate the strength of interactions. And here you use a multi-participation index, and there is a formula you sum, and you can do it for species, and you can do it also for function. I can show you the formula and the
Okay, I
equation.
think I got it. Yeah.
But it's not just sum of all the participation index. There is an index to do this. This is a product of an equation.
Okay, this is no 1 and 0. That's it.
And also these we compare to new models, and we have this compared with, it's not even maybe the final figure, But we have these also comparing the several type of models to see if it's maybe like nutrient uptake is not different from a new model, because in the island, and easy for French, we also studied this 2 times because there was a very strange result. The fungi community is super low compared, we have this result for other islands, and it's very low here. Maybe because it's a very poor soil, it's a volcanic, it's a recently island compared to the others. So maybe yes, because comparing to all the other islands that we study, these results are different, because in other islands, an Iodona, that is here in Canarias, the fungi was much more important than the other kind of fungi like herbivory. So this is a kind of also cool result, because normally fungi, they are more abundant, and even here you have 300 fungi sequences, and in the end, they are not so important because the interactions are very very weak. They are a lot, but the rest are not so strong. So here you can see that even we have like 15 types of herbivores, they have a very strong role, even being a different type of interaction. That is, this result is different for what we have found in other islands. Like here in Aradona, from the Cabrera archipelago, the fungi decomposition was the most important function. So it's not driven by the because you can talk, maybe it's driven by the abundance of the fungi, that they are much more generalist and more abundant in this community, but no, there is kind of process that is controlling these interactions, or the role of interactions in each island that is different. And we can explore more, we have more data, we have also soil data, soil analysis, or it's a kind of approach that
Marta is more passionate because she's a very fungi, obviously.
No.
But also, it's important to mention that here, like soil regulation here is interesting result also, because soil regulation here is super dry. And what I was talking with the expert in the nematodes, they say why we found a lot of nematodes in this area, because it was super dry. And they said there was interesting organisms that they stay like in sleeping for years and years. When rain's coming, they emerge again. That's why we see a lot of information for soil regulation. So in this island, it is basically fantastic to find very interesting species like slugs, caracoles, ¿cómo se llama this? Snails in this island. Snail is impossible because it's super dry for years and years and years. It was not raining, and to find snails in this island was very curious, super curious. So it's the same situation for the nematodes, or even for the fungi. Yeah.
I think we should be here. So thank you very much, Alejo.
Yeah.